So welcome everyone. I'm really excited to talk to you today about ancient DNA. I'm going to give an introduction as to what ancient DNA is, how we define it, what makes it ancient. This is a question that comes up a lot, and we'll dig into these details today. But first, I want to go back into the history of ancient DNA. When does this field start? Well, the very first paper in our field dates to 1984 and focused on reconstructing genetic sequences from an extinct form of zebra called the quagga. In this study, they retrieved two DNA sequences, which you see printed in their entirety here. So this is amazing. So today, the amount of genetic data sets that we generate are so large, there's no way that you could print all of the DNA sequences in a study. But in 1984, you could. This was an incredible amount of work to get these sequences. This did not involve digital technologies. This was entirely done through brute force biochemistry and analog methods. And what you see here is those two sequences. So on the top, you have the inferred quagga sequence, and then on the bottom, you see compared to it the zebra sequence. And so where they match, they've put a dot, and then every place where they differ, they have put the differing base so that you can see how the quagga and the zebra compare. Now, they retrieved two different DNA sequences, one from cytochrome oxidase 1, which is a mitochondrial gene. It's a fragment of that gene, and the other one which they weren't able to identify. And that's because at the very beginning of the field of genetics, we didn't have sequences for very many genes. We didn't know what they were. And so they were able to identify this fragment of DNA and sequence it, but they weren't able to confirm what it is. We now know it's another mitochondrial gene fragment. But what's really incredible about this work is that they did this before the era of PCR. So they weren't able to do what we do now, which is copy up many copies of a piece of DNA in order to make it easily visible. Instead, they kind of clipped this out of the kind of native genomes preserved within what was the muscle tissue of this museum specimen of quagga, which is really incredible. So the amount of DNA they actually needed to retrieve to do this was enormous. And the technique that they used, just really quick, one thing that is important is that this is, just to kind of put this in historical perspective, this was before a lot of digital technologies became available. So this is before Microsoft Excel, for example, became commercially available to the public. This is a spreadsheet software that today is ubiquitous and even considered a bit old-fashioned, perhaps, and yet that hadn't yet been released at the time that these ancient DNA sequences were published. So just to kind of put it in perspective, this is a very hands-on, manual, analog process. Now, the way they described sequencing this DNA, they said quagga DNA sequences were sequenced via the prime synthesis dideoxynucleoside chain termination method of Sanger et al. And that's in reference to a paper published by Fred Sanger and his colleagues in 1977. This is the so-called Sanger method of genetic sequencing that James mentioned yesterday. This was the first method that allowed us to truly sequence DNA in a kind of modern way. It is the method that opened up our eyes to the diversity of the genome. We now have many new methods of gene sequencing that are more efficient and that are faster and that are larger scale, but this was really where it all began with the Sanger method. And the way that this worked was that, as James described yesterday, you would have your template DNA molecule and you would start to make a copy of it, but you would add in nucleotides that had blocking or stopping chemistry added to them. So they would only extend a certain amount, and those would be randomly incorporated. So you would get every possible length that you could of a copy of that molecule. And you would run 4 reactions, one with blocking Gs, one with blocking As, one with Ts, one with Cs, so that they would stop at various places where these bases occurred. You could then use gel electrophoresis to order those fragments in terms of length, run them out on a gel, and then you could read that sequence. And this was completely analog. So this is just another version of that gel that I showed before on a different organism. This one's a bit cleaned up, a little bit cleaner. They ran the gel at a lower voltage over a longer time to make those bands really crisp and easy to see. But the way that you would sequence this is you would literally get a ruler and a pencil, and you would write down the sequence. So here you see we have a band in the C column, so that's a C, and then we have 2 in the A column, so it's AA, and then we have 2 in the T column, so T, T, and then a C, a T, a G, a T, and so forth. And you would build up this sequence this way, literally writing down the bases as you saw them in the gel. And so it would take a full workday to generate about 100 base pairs of sequence from performing the reaction, add in the blocking nucleotides, to then running it out on a gel. Then you would have to develop the gel. So those original nucleotides were actually radioactive. And so you would use like an X-ray to actually visualize that. Later, we developed fluorophores, which were much safer. And then you would sit down and you would write this out. And I actually, when I was very young, the very first genetic experiment I actually did involved using these old slab gels. And I learned how to use a pencil and ruler to determine the sequence. Now, this method was used to sequence, for example, this is a study from a few years later by Haas and Svante Pabo, who's a director here at the MPI and won the Nobel Prize a couple of years ago for his work on the Neanderthal genome. In his early career, this is a study they did on a Pleistocene horse. And here you see a portion of the mitochondrial DNA being sequenced using this method from this 25,000-year-old horse bone. Now, today we live in a completely different world. The amount of data that we can produce is so much greater. So today on one 8-lane Illumina NovaSeq X flow cell, for example, we can generate 25 billion read pairs of up to 150 base pairs each in just a day or two. So really, we've completely transformed the throughput at which we're generating this data. And the field has transitioned to one being very focused on biochemistry, to one being in wet lab methods, to one being really dominated by computational approaches. And that's why we're offering this course today, because today you really have to become a data scientist to be able to manage this enormous volume of data. This has completely transformed how we do science, how we do genomics, and how we do ancient DNA sequencing. So we have come an awful long way from our origins in 1984. And some might say this is why we're in this sort of data deluge. We have so much data. And so in the early days, so much of the focus was on lab work and training in biochemistry and molecular biology. Over time, those methods have become highly standardized, and where a lot of the innovation and a lot of the challenges lie is actually in the computational analysis. In the early days, the computation was easy. You had 100 base pairs to work with. You could do it with a pencil and a ruler. Today, we are no longer in that world. Today, we have terabytes of data, and that is why you guys are taking this class today to try to understand how to use these really large files. All right. But for this course, we're going to focus not on these charismatic animals like ancient zebras. We're going to talk about how we understand ancient microbes, which I think is a particularly exciting application of ancient DNA, because unlike something like a quagga or a mammoth, which you can go to the museum and you can see and you can touch and you know it's there, I think one of the most amazing applications of ancient DNA is we can go after not only these large-bodied charismatic animals, but we can actually go after things that are small, that are invisible to the naked eye. We can explore this entire microbial biodiversity of the past, from the microbiome to infectious diseases. And we can do that all with ancient DNA, something that we could never do just using something like microscopy alone. So ancient DNA technologies have really unlocked this entire microbial past. But the question I want to start with is, well, where do we get this ancient microbial DNA? Because as you know, microbes are everywhere. So how can we go, what are the bioarchives? What are the records of microbes in the past that we can reliably go to find out this information about the microbes that our ancestors would have interacted with? And there's a few reliable sources that I'm going to cover here. There are some additional ones, but I'm going to cover the major ones that we tend to focus on. The first is teeth. Teeth are an incredible source of microbes. If you had to pick one part of the human body to study, teeth are the part. They contain more information than any other part of the human body about your life. We're going to focus today on the genetic aspects of that, but you can also use stable isotope analysis, for example, of strontium or oxygen to look at mobility, or you can look at carbon and nitrogen isotopes to look at diet. You can radiocarbon date them. You can do cementum analysis to look at age of season of death. There's so many things you can do with a tooth. So a tooth is really one of the most valuable bioarchives we have of the past. Now, if we focus on genetics specifically, we can look at a tooth kind of from three perspectives. There's three main sources of microbes within a tooth. The first is one that I work on quite a lot. That's dental calculus or calcified dental plaque or tooth tartar. This naturally forms during life on the surface of the enamel. And if you have some periodontal disease like gingivitis or periodontitis, it might even start to creep down, forming on the roots of your teeth as well. This is, as I said, a natural biofilm that lives on the surface of your teeth, and it periodically calcifies spontaneously during life. It's the only part of your body that fossilizes while you're still alive, and so it's really good at preserving DNA into the deep past. What you can see here from this cross-section of a tooth, this is from a woman who died about 900 years ago in Germany at the site of Dahlheim. You can see a kind of close-up, this is a scanning electron micrograph of her dental calculus, and you can actually see these little microbes frozen in time. They calcify so quickly that they just become kind of entrapped in this matrix. And so we can recover DNA from here, and we can reconstruct aspects of the oral microbiome of this woman, plus other things that she may have been exposed to in life. Calculus entraps all sorts of things, including things like smoke debris. It can incorporate things like bits of textile fibers. It can incorporate things like paint pigments and food particles. So calculus is very valuable. The other big source of ancient microbial DNA within teeth comes from the dental pulp chamber. Now, the dental pulp chamber in archaeological context is hollow, and that's why it shows up as black on a scanning electron micrograph. But actually, during life, it is full of blood and tissue. So this is where the dental pulp is, and it's connected by the root canal to your circulatory system. So anything that's going to be circulating in your blood at the time that a person dies will also be found within the dental pulp chamber. And so if a person dies of an infectious pathogen that is bloodborne, that's circulating in their blood, such as a kind of systematic fever, something like malaria, or something like Yersinia pestis, or any other acute infection, those pathogen genomes will be found within the dental pulp chamber. Now, after death, the pulp decomposes, but some of that pathogen DNA persists, and it kind of smears along the walls of that cavity. And so we can selectively sample the walls of the inner pulp chamber to recover the pathogen DNA. And then last, during the decomposition process of the skeleton, you can see it really well in this tooth. You see this discoloration. So after this person had died, the tooth decomposed, the pulp broke down, a lot of microbes began to flourish because they had this really rich nutrient source of the dental pulp. And at first, they created a lot of acids as byproducts, which partially decalcified the tooth. But then as they ran out of nutrition, the mineral started reprecipitating, and that's why it appears extra white or on this SEM micrograph, because highly mineralized structures appear whitish, less mineralized appear gray or black in scanning electron micrographs. And so you can see evidence of that. It also looks quite ragged, and that's from where the microbes had partially decomposed some of the collagen within that tooth. And if we zoom in there in the bottom, you can actually see amongst the dentin tubules, you can see all these little tiny black dots. Those are actually living bacterial cells. So these are part of what's called the necrobiome, or the microbes from the burial environment that invade the body after death and slowly decompose it. And so while most of this happens very quickly after death, many of these microbes do persist over long periods of time and slowly break down the skeletal tissues, and we can see them there. The dentin also contains human DNA from the person's skeleton, so we often recover a mixture of human DNA and this necrobiome from such samples. All right. So in addition to teeth, where we can pick up information about the oral microbiome, infectious diseases, and the necrobiome, we can also look specifically at lesions that might have been caused by infectious diseases in the body. Now, not all infectious diseases will leave traces in the skeleton, because most infectious diseases are actually quite fast acting, and the skeleton grows very slowly. In fact, on average, your skeleton will turn over approximately every 12 to 15 years. You may not know this, but your cells are constantly tunneling through your bones and replacing with fresh bones. This is actually how you heal tiny microcracks that you form in your skeleton all the time. So you're constantly doing this process, what's called remodeling, or constantly reviving parts of your skeleton, and it turns over fully about every 12 to 15 years, although some parts turn over much less frequently, such as the petrous bone within the inner ear, which is a really excellent source of human DNA. But most of your bones will turn over rather quickly. However, it's still on a long time scale when you think about infectious diseases. So when we think about what are the infectious diseases that A, can infect the skeleton, so leaving traces, so not just the soft tissues, but B, infect a person long enough that they can actually cause changes to the skeleton, there's only a handful of such diseases. Some that are quite well known include tuberculosis and leprosy, also brucellosis, but it's a relatively short list of chronic, long-term infectious diseases that play out over months or years. I have a couple of examples of that here. So that first example comes from about 1,000 years ago in Peru among seal hunters who were later shown to have acquired tuberculosis, a form of a zoonotic form of tuberculosis from those seals that they were hunting. And tuberculosis is primarily a lung disease, a pulmonary disease, but in a subset of people, it can escape the lungs and invade other tissues. Now, it has quite high oxygen requirements, so it can't invade any tissue, but it tends to invade tissues that have high oxygen content. And that includes things like the vertebra. The vertebra have a lot of marrow in them, and so they're able to take advantage of the high oxygen levels. And so they can enter into the vertebral bodies, and there they create cavities, they destabilize and break down the bone, and this weakens the vertebra. One thing that can happen in tuberculosis when this happens is that the vertebra becomes so weak, they actually collapse forward. The bone breaks, and from the weight of the body, the person will collapse forward onto that vertebra. That process is called kyphosis, and you see an example of kyphosis here in this first vertebral column. In a person that has this happen to them, it causes a hunching of the back or a hunchback. So if you're familiar with the book, The Hunchback of Notre Dame, that kind of hunched appearance was at one time highly associated with uncontrolled tuberculosis. We also see a sculpture here that probably actually contains a mixture as depicting someone maybe with a co-infection of tuberculosis and leprosy. This person has that characteristic hunchback kyphosis, but they're also missing their limbs, which is something more common in leprosy because leprosy affects the nerves, causes people to lose sensation, and they get infections and progressively lose parts of their extremities. And then we also see a skeleton on the far right from England. This person had leprosy, dates to about 600 years ago. Leprosy also, it's caused by a related pathogen to tuberculosis, so Mycobacterium leprae. It also causes some characteristic changes to the skeleton. In addition to the loss of fingers and toes, it causes a widening of the nasal aperture, and often a loss of the alveolar bone of the maxilla, so the bone surrounding the upper teeth, and the progressive loss of the upper teeth. And so this individual who tested positive for leprosy DNA shows all the hallmarks of an advanced leprosy infection. So sometimes we can find these pathogens in the actual skeletal tissues that they infected, like the vertebral bodies or the alveolar bone, as you see here. But as I said, only a small number of infectious diseases actually make such changes in the skeleton. Hold on. Right, okay, I jumped ahead. We can also look to historic collections, which can be an incredible source of ancient DNA that can tell us about more recent infectious diseases. So things like formalin-fixed, paraffin-embedded tissue slides, for example, you can recover DNA from, and sometimes even RNA. Things like lungs that might have been preserved in medical collections in formalin or alcohol. On the bottom here, you see, I think is super cool. It's a inoculation kit. It's like a vaccination kit for smallpox, and so there's still detectable amounts of the vaccine within this kit. And so this is something I think is really exciting, and these are a bit underexplored. Don't be fooled by their recent nature. They still have a lot of damage and often have undergone different decomposition processes, but they can be really valuable sources of information about diseases, especially those that might not infect the skeletal tissues or might not be bloodborne at the time of death. Another great source of ancient microbial DNA is paleofeces. Now, paleofeces is not common, does not preserve under normal circumstances. If feces preserved routinely, we would all be like up to our ears in it. So it's really good that it does it, that it does decompose and break down. But when we do find it, it can be extraordinarily informative about lives in the past, including the gut microbiome, different aspects of health, but also the foods and diets people consumed. It can be a really rich source of information about that, especially for foods that maybe don't leave other botanical traces. Here's a couple of examples that we've analyzed in our lab. The top one is from Austria. It's a Celtic site. It's a Celtic mining site in the Alps. And the bottom right one comes from a site in West Mexico. They preserved under very different contexts. So the one from Austria preserved because it was in a salt mine, and the salt was able to dehydrate it enough to be able to facilitate the preservation of that microbial DNA. And in Mexico, it was found in a dry rock shelter, a very hot, very dry rock shelter, where again, the removal of water really helped the preservation. That's a commonality we see for paleofeces. They typically only preserve in contexts where the water has been completely removed or immobilized. So removed because it's very hot and dry, for example, or immobilized because it may be frozen in a different context. So anything that kind of stops the availability of water will lead to improved preservation. And then last, another place where we can get microbial DNA that I think is really exciting is from things like food vessels. We did a project in the Himalayas. This is in the rain shadow. It's about 4,400 meters at a site called Samsung. And there, an earthquake had exposed an ancient tomb, and we were part of the salvage archaeology project there that went to try to document it before it was lost in the seasonal melt of the Alps. And we found all sorts of really interesting preserved organic remains. So, for example, we found whole wooden bowls that still had rice, dried rice inside. We found bamboo cups full of residue. We found big copper cauldrons full of residue. All of these things represented a kind of funerary feast that had been laid out for the dead about 1,500 years ago. And so these can also be really great sources of DNA, especially when you think that in the past, food fermentation was one of the major forms of preservation. So today, we encounter food fermentation less because of refrigeration. In the last 100 years, the availability of electricity and refrigerators has completely transformed our food chain. But prior to that, pickling or other forms of food preservation involving microbial fermentation was the main means of food storage. And so a lot of foods in the past were fermented, and a lot of foods that you're familiar with today are also fermented. You may not realize it. So all bread is fermented. Of course, things like beer and wine and alcohol, but like dairy products, cheeses, most of the sauces that you might consume have aspects of fermentation. So we can also recover those kinds of culinary microbes from the past. We can also look into sediments, and this is a really exciting kind of emerging area of research where we can go into sediments and either from blocks or from different excavation techniques, we can collect sediments at different depths that can tell us about the kinds of people or animals that might have lived in a certain cave or died in a certain cave. And there's a lot of hope that we might be able to do a lot of non-invasive archaeology in the future just by looking at the decayed remains of the DNA within former habitation sediments. All right, so we've covered where you might find microbial DNA. Let's talk a little bit more about what is ancient DNA. This is a source of perennial confusion. What do we mean when we say ancient? How old does it have to be ancient? So from the perspective of our field, we don't treat ancient as an exact date, but rather we use the word ancient to refer to any DNA from a non-living source, meaning it has died, that shows evidence of molecular degradation. So we define ancient DNA by its quality of preservation, not by its chronological age. So not defined fixed age, but rather its condition. So for example, I'm going to show you some examples. So 100,000-year-old Neanderthal oral microbiome DNA from dental calculus, we would call ancient. I think everyone would agree on that. But also, for example, a 5,000-year-old hepatitis B virus from teeth, ancient. A 2,000-year-old gut microbiome DNA from paleofeces, that's ancient. 600-year-old plague DNA from skeletons, that's ancient. Oral bacteria from 19th century gorillas in a museum. We've worked with these and they show a lot of degradation, so we consider that ancient, even though it's not that old. Things like pathogen DNA from a 19th century medical specimen in alcohol. Again, we're going to call that ancient DNA because it shows the predictable forms of molecular degradation that we expect for ancient DNA. And things like leprosy DNA from a mid-20th century formalin-fixed paraffin-embedded tissue block, we're also going to call ancient DNA because it has, again, those molecular forms of damage that mean that it has to be treated differently than modern DNA. It needs different precautions in the lab, and it needs different laboratory and bioinformatics protocols. So that's how we're going to define DNA, not by a fixed age, but rather by the condition of the DNA. So let's go into more what we mean by its condition and the changes that can occur. So what is ancient DNA? So ancient DNA is damaged. It has all sorts of molecular damage that has occurred because being from a non-living source now, it doesn't have repair mechanisms to fix these damages as they arise. All right. But before we jump too deep into DNA damage, we need to know something about genomes because the different organisms that we're interested in study have very different genomes, and that has an impact on how we understand and can measure ancient DNA. So let's just do a little bit of a genome basics review. So in terms of the kinds of organisms we might want to study in the past, the first might be viruses. Viruses have the smallest genomes, but they also have the most complicated genomes. Viral genomes can come in many flavors, and I'm just showing you here one classification system for this. But viral genomes, you can have double-stranded DNA genomes. You can have single-stranded DNA genomes. So a double-stranded DNA virus might be something like hepatitis B virus or smallpox. A single-stranded DNA virus might be something like influenza or COVID-19. There's also double-stranded RNA viruses. These have a good chance of actually being found archaeologically because the double-stranded nature of it is quite protective. We have single-stranded positive sense RNA viruses, single-stranded negative sense RNA viruses. Sometimes we have viruses that have single-stranded RNA with DNA intermediates, and we have double-stranded DNA with RNA intermediates. So viruses are all over the map. Viruses are some of the most innovative pseudo-living things that play around with nucleic acids. So they're really quite diverse. And so if you're interested in viruses, you really have to know something about their genomes and how they're organized and how they're composed in order to come up with a good design. They're typically between about 5 and 100,000 base pairs long. Again, those are some of the smallest genomes that are known to exist. They can be circular, they can be linear, they can be present in single copies or multiple copies. Most viruses will just have one copy per viral particle, but keep in mind that when a virus infects a cell, it's typically not one virus, but many, many viruses can infect one cell. All right. Then we have bacteria. Bacteria are what I primarily work with, and I think within our field, this is the main focus for microbial ancient metagenomics. Bacterial genomes are a bit bigger. They tend to be between 1 and 10 million bases long. The average bacterial genome size is around 3 million bases, and they usually have one copy of genome per cell, although some will have more. They also often will have something called plasmids, which are kind of extra forms of DNA that are circular, that exist within the cell. They can often be horizontally transferred. They often contain useful genes, things like antibiotic resistance genes or virulence factors. Those tend to be between 1 and 400,000 bases long, and they can be present in one copy or up to 200 copies per cell. The other thing about bacterial genomes is that they're usually circular, but not always. Then we have animal genomes, which are more complicated and also larger. There, rather than having a genome arranged in a circular format, they're linear, which we call chromosomes, so those linear strands of DNA, and they have multiple of them. So they usually have many individual chromosomes, and those chromosomes can be present in multiple copies. So we as humans are diploid, which means we have 2 copies of our chromosomes, but other organisms like bread wheat might be tetraploid or hexaploid and have 4 copies or 6 copies. That genome is held in something called a nucleus, so we call it the nuclear genome. But in addition, animal genomes also have the mitochondrial genome, which is small and circular. It's about 15 to 20,000 base pairs long across different animals, and it's usually present in about 1,000 to 5,000 copies per cell, and that depends on the cell type. So mussels, which need a lot of energy, have many more copies of the mitochondrial genome, many more mitochondria than, say, a bone cell does. So the number of mitochondrial genomes per cell depends on the tissue type. All right. And then we have plant genomes, and plants are some of the largest genomes that are known to exist. They can be really complicated. As I mentioned, they can be diploid, tetraploid, hexaploid. They don't have as many constraints on copy number as animals do. Animals usually have very negative effects if we have additional copies of a chromosome. In fact, there's very few what are called aneuploidies, or additional chromosomes or lacking of chromosomes that, for example, in humans we can tolerate. We can tolerate sex chromosome aneuploidies, so extra copies of X or Y, and we can also tolerate extra copies of our smallest chromosome, which is chromosome 21. And when that happens, we call that Down syndrome. But most other aneuploidies that involve extra chromosomes are really can be deadly in humans. That is not true for plants. Plants can have many, many copies of their chromosomes, being completely sedentary, having a very different biological organization. They're much more tolerant of variation in their what's called ploidy, or copy of chromosome number. And so we can see really large ploidy numbers sometimes in plants. Their nuclear genome can range from about 500 million bases on the smallest end to up to 14 billion bases. So much larger than the human genome, which is about 3 billion bases in a haploid form. Typically, plants will have between 2 and 8 copies of their genomes. Again, their nuclear genome, or this genome, is organized into the nucleus, and they also have mitochondrial genomes. Their mitochondrial genomes tend to be much larger, so theirs are between 200 and 2,000 kilobases long. They usually have between 40 and 450 copies per cell. Again, that varies by the plant tissue type. And they also have, additionally, they have plastid genomes. The best known is the chloroplast genome. So this is the organelle within plants that does photosynthesis. Interestingly, both chloroplasts and mitochondria originate from bacteria that started as endosymbionts within eukaryotic cells. And so both chloroplast genomes and mitochondrial genomes are still circular. They still maintain that earlier bacterial structure to their genome. And this is called, the person who worked this out was named Lynn Margulis, and she developed the theory of endosymbiosis, or the kind of theory about the origins of the mitochondrial and chloroplast genomes. I highly encourage you to read about it. It's a fascinating story and a real success of biology of working that out for endosymbiotic theory. The plastid genomes are about 100 to 200,000 kilobases long, and sometimes they can have up to 50,000 copies in a cell, particularly a leaf cell, for example, where they're doing a lot of intensive photosynthesis. So this really matters when you're trying to think about, well, what am I going to study and how much DNA am I going to recover? It matters how big the genome is, how many copies of it is. So sequencing a tiny little virus in some ways is easier than sequencing a gigantic 14 billion base long plant genome. These pose really different challenges if you're trying to reconstruct an entire genome. Also, the copy number matters a lot. If you have just one copy or 500 copies of a particular genome per cell, it's going to really change your chance of success, depending on which genome you're trying to reconstruct. So just to kind of recap the genome sizes, so viruses are typically between about 5 and 100,000 base pairs long. Bacteria are typically between about 1, they can be up to 10 sort of, but like 1 to 5 is where most bacteria are going to fall, 3 million being the most common. Animals are typically around 3 to 6 billion bases long in terms of their genome. Plants around 6 to 18 billion is where most of them are falling, but there are exceptions. Of course, biology always has exceptions. The largest virus ever found is called the Pandora virus. It actually has a genome that's the size of a bacteria, which is amazing. It's 2.5 million bases long. The largest bacteria ever found is Syringium cellulosum at 13 million bases. Bacteria actually have pretty strong constraints on the upper limits of their genomes. This is probably pushing the very limits of what's possible, given the size of the cells and the kind of rigidity of the cells and the tension that they have in the cells. So this is probably the highest we're ever going to find. The largest animal genome comes from this Protopterus ethiopicus. I think it's a lungfish. It's 130 billion bases long. That's huge. It's like 40 times bigger than the human genome. And the largest plant genome, I think, sometimes people find plants that are bigger, but I think it's still Paris japonica, which is this tiny little plant that has a genome that's 149 billion bases long. But the biggest genome on record, still controversial because it's quite hard to determine. Can anyone guess? What do you think is the biggest genome on Earth? Anyone, any ideas? I just want to kind of drive home that sometimes complexity doesn't correlate with genome size. It is Polychaeus dubium, which is an amoeba. So this amoeba currently has the largest known genome, although it's controversial because it's so large, it's actually hard to measure. But it's estimated to have a genome size of 670 billion bases long. No one is going to be signing up to sequence this genome anytime soon. It's going to break your bank. All right. Well, I'm going to talk about a few more genome basics. I'm going to focus on the human genome because it's probably the genome that you're most familiar with. And we'll start from there and build up. So the human genome in its haploid state, meaning just one copy of each chromosome, not the two, in its haploid state is 3 billion bases long, which means because you're diploids, each one of your cells actually has 6 billion bases. We have 46 chromosomes that are arranged into 23 pairs, and they range from about 50 million to 250 million bases each. And we also have a mitochondrial genome, sometimes people abbreviate to mitogenome, which is about 16,500 bases. I think it's like 16,469. And it's presented with over 1,000 copies. If you've ever wondered why is chromosome 1 called chromosome 1 versus chromosome 2, they're ordered by length, but the naming of these chromosomes was before we had genetic sequencing technology. So it was literally based on measuring the lengths of the chromosomes under the microscope. And so you see here, chromosome 1 is almost 250 million bases long, and our smallest chromosome is 21, which is just under 50 million bases long. Turns out chromosome 22, which was thought to be smaller, is actually a tiny bit longer now that we have sequencing technologies. You can also see that the X chromosome is quite large. It's over 150 million bases long, whereas the Y chromosome is quite short. It's just a little bit over 50. And mitochondrial DNA is so tiny by comparison, it doesn't even appear on the chart. Let's take a closer look at chromosome 1. So what happens when ancient DNA is here, to remember, each chromosome is a single linear piece of DNA. That's amazing. 250 million bases, nucleotides in a single string. In order to maintain that, the cell has to constantly repair it as it acquires damage. But after a cell dies, it's no longer able to do that. So the DNA breaks into pieces. So if you think about it, if we just tried to take ancient DNA from chromosome 1 alone, it would be about a 5 million piece puzzle. It breaks into about 5 million pieces on average once it begins to decay. And how does that process happen? So this was worked out a number of years ago, and actually in the 90s and early 2000s. The first step in DNA decay is that we get something called depurination. So we get the breakage of the bond that I've shown here on our purines, our adenines and our guanines. And this causes these kind of holes within the DNA double helix. Once those holes form, the phosphate backbones become exposed and vulnerable to further chemical reactions, and they break. And this process is called nicking. This happens randomly on both sides of the DNA, and so this is what leads to what are called single-strand breaks of the physical backbone of the DNA. And then, because the only thing holding that DNA together is the hydrogen bonds between the nucleotides, which are quite weak, they tend to separate, or what we call melt. And this process of nicking and melting is what causes the fragmentation of DNA into pieces. So if you were to actually measure DNA, you'll find that on average, rather than being 250 million bases long for chromosome 1, the fragments will be closer to 50 to 100 bases long through this process. And so time is kind of acting like scissors, cutting up the DNA into tiny pieces. So how do we know when the DNA is going to melt and when it's not? Well, it all depends on the number of hydrogen bonds that you have. The more hydrogen bonds you have holding that DNA together, the more likely it is to stay intact. But if those nicks get closer and closer together, the number of hydrogen bonds holding the DNA gets smaller and they become too weak to hold it together. So something like if you have two nicks that are 90 bases apart, so you have 90 bases worth of hydrogen bonds holding the DNA together, that's likely to stay together, even in the archaeological record. So it's not going to melt. But if you have nicks that are only 6 bases apart, only 6 sets of hydrogen bonds are holding that together, that's not going to be strong enough, and likely that DNA is going to separate and melt into fragments. Just a real world example of this, it really matters because we're counting hydrogen bonds. It matters the GC content. So as you know, GC have 3 hydrogen bonds, whereas A's and T's have 2 hydrogen bonds attaching them. So the GC-rich genomes have much stronger bonding than AT-rich genomes. So just take, for example, at neutral pH, if you have a 12 base pair fragment of DNA with this sequence here, which is just G's and C's, it will have a melting point of 64 degrees Celsius. This is unlikely to melt under ordinary conditions. So if you have a kind of a 12 bases of a GC-rich fragment like this in the ground, it's going to stay together even though it's broken. However, that same 12 base pair fragment, if it's AT-rich, so entirely A's and T's, will have a melting point of about 8 degrees Celsius. That can be reached on a summer day very easily, even meters under the ground. So that is very likely to melt and separate and fragment. So the GC content matters a lot, and different bacteria have different GC contents, which will affect their relative degradation rates. All right. The next thing that happens is once we have those single-stranded overhangs, the cytosines become quite vulnerable to deamination. So you can see here the, my cursor. These nitrogen groups here become vulnerable and they can be removed. And when that happens, they get converted into uracils. So the thymine becomes, or the cytosine becomes a uracil. Uracils are not normally present in DNA, they're components of RNA. A lot of polymerases, though, don't recognize uracil, but misinterpret it as a thymine. And so this is why when we look at ancient DNA, if we look at the patterns of misincorporation or unanticipated bases at the edges of reads, we often see this increase in the number of thymines on the read termini, and that's because of the excess of uracils that have formed on these single-stranded overhangs. So just a recap of DNA damage. First, we have depurination. This is the random loss of A and G bases. This leads the DNA molecule to become vulnerable to nicking, or this hydrolytic attack of the phosphate backbone at the sites of depurination. Then we get fragmentation. So when two of those nicks on opposite strands are very close together, the hydrogen bonds between those bases aren't strong enough to hold the strands together and they separate or melt. And this causes fragmentation with single-stranded overhangs. And then finally, we have deamination. Cytosines on these single-stranded overhangs undergo a hydrolytic attack and lose their amine group, and this converts them into a uracil. DNA polymerases will then read the uracil as a thymine, introducing these cytosine to thymine errors in downstream sequences. All right, so if you think about DNA as being like this wine glass, what you get with ancient DNA is this kind of shattered remains of it. And so if we think about now applying this not to the human chromosome 1, but to bacterial genomes, with their smaller genomes on average of about 3 million bases, it means that a typical ancient genome from a bacteria is going to be broken into about 60,000 pieces. It's a slightly better puzzle, but still very hard. So let's go back and figure out, like, how was all this figured out? We want to put this genome back together, but how was it figured out that this happened? So in the pre-next generation sequencing era, this was in the era of kind of PCR, we knew that ancient DNA was fragmented, but we didn't have any way of actually measuring the native fragment length distribution of those ancient DNA reads. We couldn't measure it precisely, and for a lot of reasons. So short DNA is really easily lost during extraction. DNA recovery was too low to visualize on a gel. So all the tools and techniques we had in the pre-next generation sequencing era really prevented us from understanding the nature of DNA that was happening. So you'll find in the literature from this time lots of guesses of ancient DNA being around 100 to 500 base pairs long. It was sort of an educated guess, but couldn't be precisely measured. Early PCR designs often tried to target things that we now know were way too long. So 300 to 500 base pair fragments, which often don't exist in ancient DNA. And this led to a really high PCR failure rate and also tons of contamination problems because it was much better at amplifying contaminant DNA than truly ancient DNA. It was also known for some time that there always seemed to be an excess of C to T miscoding lesions and also G to A in ancient DNA, but it really wasn't understood why that was happening. And at this time, DNA damage was really just seen as a problem that no one quite knew how to overcome. This all changed in the next generation sequencing era. So instead of requiring primer sites on the DNA template, what NGS did was it ligated universal primers onto the ends of all the molecules. And this made it possible for the first time to recover all of the DNA in a sample and to measure its true size, which was revolutionary. It also allowed us to determine the order of damage processes so that the process of DNA degradation could be defined. And a lot of this was worked out between about 2007 and 2014. There at the time in 2013 or 2012, there was a new DNA extraction method that was developed that allowed us to recover these very short fragments. And this really revealed for the first time just how short ancient DNA really is, and often has an average fragment length of about 30 to 50 bases. And because many of these processes are actually quite predictable, this new way of analyzing ancient DNA turned this into a kind of solution for authenticating DNA. It was no longer just a problem, but we could take advantage of our knowledge of these processes and show that they were occurring in samples we were interested in to actually authenticate it. So these very things that had been seen as a disadvantage were now a strong advantage that we could use. So in 2007, some of these first observations were made, and one of them was that the reads always seem to break right after a purine. And so they disproportionately, the read break would happen if you were to map your DNA back just outside of the breakages, you would find purines. And that was one of the first kind of evidences that we had that depurination might be what started this. Also, once we were able to get next generation sequencing data, we could look at the damage on the ends and show that these uracils were really concentrated excuse me, on the ends of the molecules where we have these single-stranded overhangs. It turns out that cytosine deamination happens about 1,000 times faster on single-stranded overhangs than it does in double-stranded DNA, which is why we always see an excess at the ends. This plot has been nicknamed the smile plot because of this characteristic shape. And it's caused again by the randomness of this nicking. So this time shears up our DNA into pieces, just like using a shearing instrument does. All right. And it's one of the reasons we see the C to T on one side and G to A on the other, which is very typical of double-stranded DNA library constructs, has to do with the asymmetric behavior of repair enzymes during blunt end library construction. So I'm just going to go through this a little bit here. So it's really important to know that DNA has a 5' to 3' orientation. So it runs in one direction, and then the opposite complementary strand runs in the other direction. So I'm just going to draw this out here. And we use something called T4 polymerase to repair the DNA as we prepare it for attaching on the kinds of adapters that make it readable by a next generation sequencing instrument. So let's just talk about what happens here. Why do we get this smile plot with C to T's on one side and G to A's on the other? How does this occur? So the first step of next generation library construction is to repair the DNA, because we can't sequence DNA that has all these overhangs. We need to make them blunt. And we do this with an enzyme called T4 polymerase, which ultimately comes from a virus. What T4 polymerase does, cuts off the 3' overhang. So anytime you see a 3' overhang, it just trims it off, just cuts it. And then it extends the 5' overhang. So T4 polymerase has this orientation. So it cuts off the 3' overhangs and extends the 5' overhangs. And when it does, it puts in the complementary base opposite the uracil, which it's going to interpret that uracil not as a cytosine, but as a thymine. So it's going to put an A opposite it. Later, when you go to melt those strands, which you do before sequencing, if you reoriented them all, what you can see is you have uracils all on the 5' end of the molecule, and you have their complementary adenine now on the 3' ends of the molecule. And so this is what leads to that characteristic C to T and G to A pattern. It's an artifact of this asymmetric behavior of the T4 polymerase. All right. The 3' did have T's. They were just all cut off during the repair step. All right. So fun fact, because this damage typically only occurs on single-stranded overhangs, the misincorporation rate can usually never reach 1, and the maximum rate under normal circumstances is 5. It's 50%. All right. So we can use, there's a number of different tools now that have been developed that allow us to actually really rapidly measure these known forms of DNA damage, including map damage, PMD tools, damage profiler. We're going to use damage profiler in this class because it's quite fast and it's comparable to the others. So we can look at these as these patterns of DNA damage as an authentication tool. So when we see no uptick of thymines on the ends of the molecules, for example, or adenines, we have this sort of not ancient signal, and when we do, we have this ancient signal. Sometimes people will see this other pattern here where it's really spiky. That's common if you don't have enough sequences to actually statistically run the model. You typically need at least 1,000 reads to do this. And sometimes you get the correct shape, but the baseline is elevated, and that's a good indication that you've used the wrong reference genome. A lot of people want to know, can we use damage as a clock? And the answer is sort of, but not really. It's more like a clock that says only today or a while ago. Here's an example of what I mean. The relationship to time is not linear, but is really dependent on local temperature and humidity. So here you can see in green is some data from 5,000 years ago in Scandinavia, and it has terminal deamination rates of about 25 to 30%. And you can see in black is a Neanderthal that's 40,000 years old, and it has higher rates of DNA damage that are around 40 to 45%. So that's great. But then you see an example here from Costa Rica, which is the youngest of all the samples, only 1,000 years old, and it has damage rates that's much higher at 50%. So within a given environment, DNA damage accumulates time in a predictable way, but in different environments, that clock could speed up or slow down. So you can't use it as an absolute clock. It also will vary by organism, even within the same sample. So we've measured multiple organisms within the same skeleton, for example, a pathogen, a human, for example, and their DNA will degrade at slightly different rates because of the different properties of their genomes and also the kind of tissue type in which they are. There are some ways that we can remove this damage. For example, if we want to have really clean genotyping, it is possible to remove. And this is often done using user treatment, which stands for uracil DNA glycosylase combined with endonuclease 8. And what this does is it allows you to, the first thing you do is you phosphorylate everything with P and K, and then you add the user treatment. And anywhere where a uracil occurs, it just cuts it off. So it clips out the uracil base, leaving an A basic site, and then endonuclease 8 comes and clips it at that position, which shortens the DNA. And then T4 polymerase comes and trims the 3' overhang and fills in the 5' overhang. And now your cytosine damage is now gone. So you can remove the damage using this process, but it's really by clipping out the damage. So you end up with a smaller molecule. Can you have your cake and eat it too? Can you remove some of the damage, but not all of it, so that you can still see a little bit of it, but maybe not have it extend so far into your molecule, because that can make it really hard to be confident in your genotyping? And the answer is yes. So a new technique has been developed called partial UDG treatment or UDG half treatment. And in this case, it leaves damage only on the outermost base. So then you can very easily say, ignore the last base, but you can still see the damage to authenticate it, but you can clip that off for your analyses so you can be really confident in your genotyping. The key here is phosphorylation. So basically the full UDG, you first use P and K to phosphorylate the ends, and then you use user treatment to clip out the uracils. And so you remove it all. What you do with UDG half is that you just don't add. You just don't add the P and K. It does not phosphorylate. And then user treatment can only affect uracils that have been phosphorylated. So internal uracils are all phosphorylated, but the extra ones often aren't. And so what happens is it will only clip off the internal uracils, leaving any that are at the end unchanged. And so when you go to sequence that, you'll only see uracils on the terminal base. What that means, however, is that you'll only see a small portion of the damage. So the damage levels are always lower in partial UDG treated libraries. All right. So everything we've talked about so far is valid for DNA sequence data generated from standard double-stranded DNA libraries, but increasingly people are also using what are called single-stranded libraries. And this is more efficient for recovering shorter amounts of shorter DNA, and also for it often can recover more endogenous DNA. In this one, you do not clip off the 3' overhang, so you keep all of your original damage. All right. And as a result, for single-stranded libraries, you're going to see that C to T misincorporation on both sides of the molecule. So you're going to have a little bit of a different damage pattern. So the reason why people like to use single-stranded libraries, it avoids a problem that we have with double-stranded libraries. So with double-stranded libraries, we have to first blunt in our DNA through this DNA repair, and then we ligate on our adapters. But the problem is we don't have control over this ligation process. So sometimes you get the same adapter, like the P5 on both sides. Sometimes you get the P7 on both sides, but what you really want is the P5 on one side and the P7 on the other. So when you do a double-stranded library build, because of this blunt ending a ligation step, you lose half of your library to improper ligation. And so 50% of your library is gone. Single-stranded library preparation works differently because you melt those strands. Every single molecule gets built into a library. So this can really increase your recovery of ancient DNA from really low biomass ancient samples. So just as a quick damage wrap up, we've got over here on the first one, we have no damage. So this means it's either modern DNA or it's a full UDG treated library. We have this spiky pattern, which means you actually didn't have enough DNA to run this analysis. It's not statistically valid. Go back, get more DNA. You have this one where you have a good pattern, but the base is elevated, so it means you have the wrong reference genome. Maybe try a better one. This one is okay. This looks good if you did a partial UDG treatment. This one here, we have C to T on both ends of the molecules. This is what you would expect for a single-stranded library prep. And this one is a beautiful double-stranded DNA damage protocol. And I think, James, I'm going to check in with you. On my timer, I still have a few minutes, but you seem to have a different timer. So I'm just going to try to wrap up here really quickly. All right, so the last thing I want to cover is about enzymes. This is something that a lot of you talked about, wanting to know more about wet lab work. Wet lab work matters a lot because there's many different enzymes and chemicals that people are using in the lab that can have huge downstream consequences. So one of the most important ones is the choice of polymerase, DNA polymerase that you use in your library prep. So, as I mentioned before, one of the characteristic forms of damage that happens in ancient DNA is the cytosine loses its amine group, which converts it into a uracil. Uracil is not a normal component of DNA, it's a component of RNA. And so one of the problems is that different polymerases treat uracil differently. So far, we've discussed how enzymes like T4 polymerase treat uracil like a thymine. And this is how you get these introductions of C to T misincorporations. But not all enzymes do this. Some enzymes, when they encounter a uracil, just stop. If that happens, that molecule will not end up in your sequencing library and will not end up in your data set. So the damage present in ancient DNA, the fragmentation and deamination, requires the use of specialized library protocols specifically for ancient DNA. So DNA polymerases come in two flavors. Non-proofreading, they treat uracils like thymines. That's going to be things like your Taq polymerase, that's going to be your T4 polymerase. And then you have things that are proofreading, that stop at a uracil. Because for a lot of modern DNA, they have so much DNA, they don't want to deal with uracils, so they don't even want to see it. And so people have developed these proofreading enzymes, which actually have a high fidelity and have all sorts of advantages, but a terrible disadvantage for ancient DNA. So for ancient DNA, it's critical to use a non-proofreading polymerase for library construction and the indexing PCR in order to lock in that damage by turning the uracil into a thymine. Once that's done, for downstream steps, you can use your proofreading enzymes, but that first one must be non-proofreading. If you use a proofreading enzyme for library construction, say by mistake, your damaged ancient DNA molecules will not be sequenced, and that may bias your data set towards contamination. However, a UDG-treated DNA library is compatible with proofreading enzymes because the DNA damage was removed already. All right. So why use proofreading enzymes at all? Because they're more accurate. And so if we use proofreading enzymes for every step except those two key steps in which the polymerase encounters the original damaged cytosines. So we use the non-proofreading T4 polymerase for DNA repair. We use a non-proofreading polymerase, something like PFU Turbo CX for library indexing and amplification. And then everything downstream of that, all the subsequent amplifications, the reamplifications, the reconditioning steps, those can all be used using a proofreading enzyme, something like Hercules 2. If you want to know more information about this, you can visit our online bench protocols for ancient DNA sequencing and all of our laboratory protocols on protocols.io. So let's take one last step back and say, what is the big picture? Why does DNA damage matter? DNA damage is important because it allows us to authenticate individual species or metagenomic assemblies or even individual reads. However, it can pose major challenges for taxonomic identification of sequences, accurate genome mapping, and metagenomic assembly. But it turns out the biggest challenge in our field is not the cytosine deamination. We can actually manage that pretty well. The biggest problem for us is fragment length. Sometimes our DNA is just too short. And the reason for that is that taxonomic identification of sequences requires enough bases to be able to make a relatively unique match. DNA fragments that are less than 30 bases long lack sufficient specificity for taxonomic assignment. They align to too many genomes with no phylogenetic coherence. And that is really why we have limits on the oldest DNA that we can find. It's not that DNA magically disappears at 3 million years ago, which is the projected limit of how far we can go back. That's the time it takes under ideal conditions to make all the DNA less than 30 bases long. DNA sequences with less than 100 bases are difficult to assign to a taxonomy. They often lack the taxonomic specificity within clades, and this can lead to cross-mapping or false mapping with related taxa. When there are insufficient reference genomes for a given species or a genus, these short sequences can easily be misaligned to the wrong strain or species. This can often happen when you have some sort of maybe harmless commensal bacteria that's related to a pathogen, but because that commensal is not in a database, you may map to the pathogen genome and falsely believe you have a pathogen when actually it's a harmless relative. This can also cause big problems for genotyping, building phylogenies, and inferring evolutionary histories. So one solution that the field is increasingly turning to is metagenomic assembly, and this is where we don't rely on those incomplete databases of reference genomes, but instead try to reconstruct the genome simply from our own data. It can be difficult to do de novo assembly with sequences that are less than 250 bases long, but there's increasingly improvements in our methods that allow us to use the short fragments of DNA to do this. One of the reasons it's difficult is that if you have really short sequences, it can lead to lots of short contigs. And the reason for that is that they aren't really long enough to span repetitive elements or other things. So you end up with kind of smaller contigs or contiguous assembled sequences. And many assemblers automatically discard short sequences. So you have to be sure to change default settings. And there's a number of ancient DNA papers that give you guidance on what specific settings you need to change in order to make it work for your ancient data. Metagenome assembled genomes, or MAGs, are possible, and they're increasingly becoming the workhorse of our field and are leading to amazing discoveries. But you have to fine-tune your pipelines in order to get them to work properly on ancient DNA. So as my last recap on ancient DNA review, ancient DNA has changed enormously since its beginnings in the 1980s. Gone are the days of radiographic films and rulers for ancient DNA sequencing. We now have machines capable of churning out 10 or even 25 billion sequences at a time. And this means archaeogeneticists today must learn coding and scripting. And so we're really happy that you're in this class to learn that here. Genomes are big, but they fragment into thousands or millions of pieces once an organism dies. And the shortness of these DNA fragments with a kind of mode length of 30 to 50 base pairs with a max of about 150 base pairs can make taxonomic identification, genome mapping, and metagenomic assembly hard. Ancient DNA accumulates damage, but we can characterize this fragmentation and cytosine deamination as indicators of authenticity, but not as a precise age. And ancient DNA requires a specialized laboratory and library protocols in order to handle this damage. And we now have options to remove the damage with UDG, or we can recover even more damage with single standard library protocols, depending on the application. DNA fragmentation is currently our biggest challenge in the field.